If you were here a couple of talks ago, Torsten did a wonderful job of introducing Potentilla in general, so I'll talk a little more about just this specific group. I studied the Potentilla breweri complex, which, depending on who you ask, is somewhere between 5 and 12 species of cinquefoils, mostly from the Intermountain and Northern Rockies, the Western North America. It's a small subset of a big genus, that number of species out of 400 or more. It includes Potentilla breweri, Drummondii, Brucei, Amicarum, recently described, versicolor, Potentilla millifolia variety algida, which I'll explain that in a bit, and a number of synonyms and undescribed groups that are closely related but currently lack names. There, as you can see, unassuming little high montane to subalpine perennials with leaves pinnate to subpalmate. What did I just do? Oh, for Pete's sake. Sorry about that, folks. Pinnate to subpalmate leaves, yellow flowers, 5 petals. I'm pretty sure even their pollinators can't tell the flowers apart, though they have not been terribly useful for systematic purposes within this group. Again, montane to subalpine, primarily in shortgrass meadows, rock crevices, and areas similarly low on competition for light, all of them seasonally very wet. About 7,500 to 11,000 feet elevation, which is around 2280 to 3350 meters. And again, Western North America. The farthest inland points here are likely identification errors, but Potentilla Drummondii's range covers pretty much all the territory that the other groups' ranges do. As you can see, it gets all the way into Alaska. The rest are mostly in the US end of the range. Because these little critters are so picky about habitat, needing such little competition for light and high moisture, even in the high Sierra Nevada, which is where their habitat is most continuous, it's pretty patchy. In the Great Basin, we're talking outright sky island distribution. And within that habitat, hybridization is absolutely rampant, both with each other, members of the group, and with several other Potentilla found in sympatry. I often encounter hybrid zones like the two here, Bigelow Lakes and Baldy Mountain from Northern California and Eastern Oregon, respectively, where you'll find fairly uniform populations of a pair of parent taxa, breweri and Drummondii, or versicolor and gracilis. These in fairly large numbers, consistent populations, and this smear of intermediate hybrids, which quite frankly should not work. They should be blended together. Facultative apomixis seems to be the likely explanation, that they only occasionally reproduce sexually and are otherwise cloning themselves. These plants have been giving taxonomists headaches since at least the 1940s. Klassenkeck and Heise used breweri, Brucei, and Drummondii as part of their seminal field-defining biosystematic experiments. The results were telling, but somewhat taxonomically ambiguous. They ended up putting Brucei and Drummondii into a single species, but found enough plasticity that there was still some blur going on there. They also found frankly unreasonable chromosome counts, which I'll get to in a minute. Molecular work has not been very much more illuminating yet. The best published work, at least last I checked, seems Torsten's group is working on some very exciting things around here. Only the Dobish and Paul paper only sampled one member of this group, and of course it fell squarely into this Argentia clade C, which is one giant polytomy, effectively. So, this pattern of diversification is not straightforward. I'm working on making sense of it. So far, the project has mostly consisted of sampling, lots of sampling. I've collected the plants from all over the range of each species, mostly, again, California, Oregon, and Nevada, where the interesting stuff happens. Picked up about 250 specimens so far from the Wallowa Mountains to the Sequoia National Park in the southern Sierra Nevada, and from the Ruby Mountains to the western Siskiyous near the coast. For every subpopulation of each species present, I've been getting at least one voucher specimen, DNA, leaf material and silica gel, seeds and or live transplants also wherever possible. From there, I'm going to be building a well-supported, ideally, molecular phylogeny. Thanks to rampant hybridization, I cannot assume this is a tree. And thanks to the apparent fact that the little creatures trade chromosomes like baseball cards, I also cannot use Sanger sequencing and expect to get any portion of the whole story. Next-gen sequencing, targeted sequence capture specifically. I'm currently waiting on my first set of anchored phylogenomic results any day now. I'm also going to be doing some Sanger sequencing just to pull in previous previous sequencing data. Some unpublished results from Matt Williams, who worked on general Western North American Potentilla, and of course GenBank. These are my extremely rough Sanger sequencing preliminary results. Most of these were pulled from GenBank. The bright green, the bright green are the ones I've actually sequenced, and as you can see, I'm just getting more of that giant polytomy. Really looking forward to the next gen results. As I mentioned before, the chromosome numbers are frankly unreasonable. Klaus and Keck and Heisey found, well, this. The one that always gets me is Potentilla brucei, 63 through 73 inclusive, and yes, these are diploid numbers. I suspect a lot of univalent chromosomes. Flow cytometry of the living plants that I've been transplanting has gotten me some good C values, which, predictably enough, are large and highly variable. I've got a 6-fold range of variation here. Facultative apomixis, again, is the possible explanation because it mitigates reproductive failure risks of ridiculous chromosome numbers. If you cannot undergo successful meiosis, you can still reproduce effectively asexually using apomixis. In some ways, you could consider it sexual reproduction writ large, just like just as recombining gametes does in the first place. It lets you keep the genotypes that do work while still mixing things up enough to maintain variation to be able to adapt and or deal with parasites. The 4 morphologies I mentioned before is little to no help, other than the styles. Neither experts nor their pollinators can easily tell this group apart. The vestiture, especially of leaves and petioles, however, is key, rather diagnostic. The degree and type of pubescence is often mixed. You'll get these stubby little glandular hairs, along with maybe an undercoat of short, crisp hairs, along with longer, silky ones on top. So I am looking forward to doing some morphometrics. Well, I can't say I'm looking forward to counting individual hairs, but the results will be interesting. The shape of woody caudex that's found at or just below the soil surface is also worth some study. Some are branched, some are simple. It's currently a more or less binary character, but we'll see where that goes. The inflorescence branching patterns are cymose, as pretty much everything in Potentilla is. Used in previous treatments in fairly qualitative terms of openness and less qualitative branching angle. I'm looking for morphometric ways to handle the whole branching structure. Persistent homology looks promising, but still working on that as well. And then this is where we get weird. The leaf shape and division, as you can see, is highly variable and somewhat irregular. Just the shape of, say, a terminal leaflet within a species gets all over the place. Most leaves are pinnate, or at least pinnate palmate intermediates, a pinnate leaf with exaggerated basal leaflets. But again, there is substantial variability to the point of, from this particular Steens Mountain specimen, pinnate leaflets, leaves, and palmate leaves on the same plant. Next gen geometric morphometrics has enormous promise in teasing apart similarly ambiguous and hard to define shape variability. My lab has particularly been using elliptical Fourier morphometrics, in which you take the elliptical Fourier transform of outline shapes and do a PCA of the resulting distribution of ellipses. It has enormous power to tease apart just this kind of ambiguous shapes. Unfortunately, the algorithm itself can't handle self-overlapping shapes, which rules out using the outlines of whole breweri complex leaves. The math exists for 3D elliptical Fourier, but as far as I can find, has not been implemented in something I could actually use, short of becoming extremely fluent in several programming languages. What I've been doing instead is taking just a terminal leaflet. Here's a simple panel of terminal leaflet shapes. The lower resolution ones were pulled from floras, the higher resolution from images of, from photos of common garden plants. For example, this pink alone, those are all from a single Potentilla versicolor plant. As you can imagine, the PCA has not yet been as informative as I'd like. And then once I do manage to actually quantify and compare these shapes, there's the issue of plasticity. How can you compare results from plants sampled in dramatically different habitats as dramatically different as the, say, the meadows at Timberline, and which the grasses are a couple decimeters tall, and alpine fell fields in the Wallowa Mountains, where if anything gets above a couple centimeters, it's unusual. To deal with that, I've been building a common garden. As I mentioned, I've been acquiring live transplants. They're currently growing in a cold house at University of Wisconsin-Madison, where I'm from. And I'm looking to quantify these morphologies in the same ways I'll be doing the rest of them, and compare to the actual wild vouchers to build a model of response. The garden is currently getting well established. I've had, I think, 7 separate plants bloom this year, but has a bit of a ways to go. So in conclusion, this project is not quite in its infancy, but still very much in development. But the goal is to use a broad, integrative approach across several systematic methods to resolve the systematics of a historically very difficult group. Yes, it's ambitious. I recognize that I'm breaking a few lances against the local windmills. But if I actually can resolve this, the methods will hopefully suggest the results will hopefully suggest ways to integrate these methods to apply to other historically difficult groups, which areas are most likely to successfully co-illuminate. They should also tell us what the natural groups and natural units of diversity are, which will be valuable in conservation work for these sensitive species in sensitive environments. That, of course, leads into the general conservation activity. Sorting out the biogeography and especially the post-Pleistocene history of these plants will help figure out how we can help them through the Anthropocene, in which, well, with climate change and specifically warming, they are rapidly running out of mountain to climb. They're not alone either. Again, sampling is underway, analysis is in progress. And besides all these useful applications, this does get me out in places like this every summer, which I'm not exactly inclined to call a downside. Thanks for listening. I have had a lot of help with this project. A number of people to acknowledge, not least the NSF, the University of Wisconsin-Madison, and the University of Jefferson-Bavaria at UC Berkeley for funding. Any questions?
Time for one.
Alexa, do you have reason to believe that the chromosome number changes, the chromosome arrangements within breweri are preventing gene flow between populations, or is it not that?
On the one hand, yes. On principle, I'd expect something with 63 chromosomes and something with 73 chromosomes to find interbreeding impossible.
What are there intermediates where there's just one chromosome rearrangement, one chromosome rearrangement, so the genes can hop around between populations?
That's what I'd expect, given that they do appear in the field to be trading genes despite the chromosomal incompatibilities. Sorry?
But it's not known yet.
It's not known yet. I'd very much like to get chromosomal data, but karyotyping is more time consuming than my committee wants to spending much effort on, and flow cytometry doesn't really give you ploidy level when you have no idea of chromosome size.
Thank you, Alexa.